of Eastern Medicine, Government College, University, Faisalabad. I will discuss significance of transaminases. They help in the formation of non-essential amino acid, alanine and aspartate. Alanine is converted to pyruvate, which is a substrate for gluconeogenesis. Phosphotransferases, they catalyze the transfer of phosphate group, also called kinase. Glucose is converted to glucose 6-phosphate in the presence of hexokinase and glucokinase, and ATP is converted to ADP. Transmethylase, they catalyze the transfer of methyl group. Noradrenaline is converted to hippuric acid, and enzyme is transmethylase, and S-adenosylmethionine gives the methyl group. Transpeptidase, they catalyze the transfer of peptides or amino acids. Benzoic acid is converted to hippuric acid, and transpeptidase is enzyme. Glycine is amino acids that combines with the benzoic acid and hippuric acid is formed. Transacylases, they catalyze the transfer of acyl group. Palmitic acid is converted to palmityl esquate in presence of transacylase, and coenzyme A is used. Hydrolases, they catalyze hydrolysis, breakdown of a compound by the addition of water into the substrate. Protein hydrolyzing enzymes, carbohydrate hydrolyzing enzymes, lipid hydrolyzing enzymes, deamidases, and other ester hydrolyzing enzymes, phosphatases, and miscellaneous. Thank you very much. Hydrolyzing enzymes. Protein hydrolyzing enzymes, also called proteinases, proteases, or proteolytic enzymes. It has two substrates. Exopeptidases, they act on the terminal end of the polypeptide chains. They are further divided into dipeptidases, tripeptidases, and polypeptidases. Aminopolypeptidases, which are present in standard use, act on the amino group, amino terminal of polypeptides. Carboxypolypeptidases, which are present in pancreatic juice, act on the carboxy terminal of polypeptides. Endopeptidases, they act on the central peptide bond of the polypeptide chain. They include pepsin present in gastric juice, trypsin, chymotrypsin present in pancreatic juice. Carbohydrate hydrolyzing enzymes, carbohydrates catalyze the hydrolysis of glycosidic bonds, amylase, maltase, sucrase, lactase. Starch is converted to maltose in presence of maltase, amylase, and maltose is converted into glucose in presence of maltase. Lipid hydrolyzing enzymes, they are of the following types: lipases, they break triglycerides that are also called neutral fats to release glycerol, fatty acids, and mono or diglycerides. Lipases or lipase is mainly present in pancreatic juice, and thus pancreatic lipase is very important in fat digestion. Now you can say there are the two organs that are important in the body for the digestion of the lipid: pancreas and bile juice. Pancreas and bile juice is involved in the emulsifications of fat. They emulsify the fat, and emulsification is important part in the digestions of the fat. And the pancreatic lipase is enzymes that hydrolyzes the lipids. Emulsification is done by bile and digestion is done by pancreatic lipase. This is very interesting thing. Cholesterases, they hydrolyzes cholesterol esters. Phospholipases, previously called lecithinases. These are of four types: A, B, C, and D. Phospholipase A is present in certain snakes' venom. It forms lysolecithin, which is a strong hemolyzing agent. It means when it is present in the snake venom, and when the snake cuts the human body and hemolysis occurs, and as a result, the person dies. Deaminases are amino hydrolases. They remove amino group from an amino acid in the form of ammonia and convert that amino acid into its corresponding keto acid. For example, glutamic acid is converted into alpha ketoglutarate in presence of deaminase, and water is converted into ammonia, and amino group is obtained from glutamic acid, and glutamic acid is converted into keto acid, that is the alpha ketoglutarate. Deamidases, these are also called amidohydrolases. They remove an amide. Amide means NH from an amino acid in the form of ammonia, asparagine, that is converted into aspartic acid, and ammonia is released, water is entered. Deamidase is enzyme. Other hydrolyzing enzymes, there are two groups, phosphatases and miscellaneous, and we will discuss it later. Thank you.